high-level webinar series, in particular to Professor Luciano Sasso from Sapienza University of Rome, that spearheaded the webinar series. A special gift to Luciano that are unable to be with us today, and we are grateful to Antonio for kindly leading this webinar. Now it's my pleasure and honor to give the floor to Professor Antonio Quadrato for his welcome.
Thank you very much, Brigitta. You are very kind for your words. And I also want to thank the people who are connecting from many places. We are really very, very happy on the cost action that we have been running for almost 4 years now. One of the activities that we had was this webinar series. Today we have the 8th webinar. I think they have been very successful in connecting people all over Europe, mainly Europe, but not only Europe, and also spreading the word about this wonderful transcription factor called NRF2. Terrible name for a wonderful transcription factor. Anyway, I think this is all I want to say. Just thank you again. And maybe what we can do is to start all over with the specific webinars. Maybe, Brigitta, would you like to give just a brief comment on how we are going to organize this?
Yes, I want to remember that each speaker will give a talk about 20 minutes. The webinar will last up to hours. The discussion will be open after all the talks. Please feel free to ask a question in the chat at any time. And when doing so, please don't forget to mention your name and affiliation to whom the question is addressed. Of note, the webinar will be streamed online upon registration. So we can start with our program.
Okay, so maybe we can start with our first speaker today. Our first speaker is Professor Nikki Kondogami from the National Genetic Research Foundation in Greece, in Athens, I believe. And she has been working on this very nice connection between NRF2 and the proteasome. And I think she's going to talk about the relevance of this axis in aging. So, Nikki, please, whenever you want.
Indeed. Thank you. Thank you very much for the kind invitation. Let me share. Do you see my screen? Good.
Yes, we can.
Good. So thank you, Brigitta, for the nice invitation. Thank you, Antonio, for everything you have done so far with BenPetFar. I'm working at the National Hellenic Research Foundation in Athens, and I'm working with aging, and I'm going to show the effects of NRF2 modulation on aging, health span, and longevity, sharing also results from other groups. So aging is characterized by a gradual accumulation of alterations in the cellular and organismal physiology, and this gradual accumulation eventually leads to disrupted biochemical pathways, to increasing damage, and at the end to the failure of cellular and organismal homeostasis. Now, we all have seen people that we say they look younger than they are or they look older than they are. This is exactly the difference between the chronological age, which is just a number of years a person has lived, and the biological age, which is the true age of a person. And nowadays, we know in the aging field that we all age in a different rate, and the idea is whether we can decelerate this rate in order to have a lower biological age, and we don't care about the chronological age at all. This also brings us to the difference between the lifespan and the health span. So lifespan is the total number of years a person shall live, whereas health span is the total number of healthy years. So more or less, we reach the age of 85, but already from the age of 50, we start accumulating age-related diseases. However, if you see the example of centenarians that are considered as a successful and healthy aging example, you will see that not only they have accumulated 15 more years of life, but if they start accumulate age-related diseases, they do it at the age of 85. So we appreciate that they have accumulated 35 years of healthy aging. So in the aging field right now, we are more focused on whether we can extend the health span. And of course, if lifespan comes, this is welcome as well. So a few years ago, and 2 years ago, they were updated, we had the hallmarks of aging. So I tried to see which one of them is directly or indirectly linked to NRF2. And you will see that in all of them, you have implication of NRF2. So it's not really a surprise that for NRF2, it is suggested that is the master regulator of the aging process, the guardian of health span, or the gatekeeper of species longevity. This is what has been suggested. So the protective role of NRF2 signaling comes from the activation of transcription of different genes related to detoxification of drugs and toxin, protection against oxidative stress and inflammation, but also it plays a huge role in stability of proteins and in the removal of damaged proteins through the proteasome or autophagy. The first studies that came regarding NRF2 came in the nematode, where NRF2 is skin1, this is the ortholog, and it was shown that if you modestly overexpress skin1 in the nematode, you have a lifespan extension. So you can clearly see in black, this is the wild type, and in the colors, the lines in the colors show different transgenics overexpressing skin1. Now, if you kill skin1 in C. elegans here by the green line, so if you have mutations or if you silence skin1, then not only you don't have the lifespan extension, but you have lifespan shortening. But the catch is that if you overexpress skin1 from high copy arrays, then you don't, it's not like you can extend further the lifespan, but you have toxicity. Moreover, many of the people that have overexpressed modestly skin1, they have also tried to see what is happening to the health span of the animals. And one of the markers that you have to see whether the animals are more healthy, it's to see their movement. So you can clearly see here that the animals that modestly overexpress skin1, they maintain their motility for longer and they are healthier in that way. So in the nematode, it has been shown that modest overexpression leads to lifespan extension and health span amelioration. If you move to Drosophila, where you have CNC NRF2, then you can see that people have overexpressed NRF2 through KEAP1 loss of function mutations, and especially in the male animals, you do see an extension of lifespan when NRF2 is enhanced in that way. However, if you hyperactivate NRF2, either very early in the lifespan of Drosophila or constantly, not only you don't have lifespan extension, but you have accelerated aging, showing again what it was shown in the C. elegans model. And of course, if you see the health span, you can see that NRF2 activation enhances health span. In Drosophila, when you put them in a bottle, they are starting climbing. The higher they go, the healthier they are believed to be. This is the negative geotaxis. And you can see here when you silence KEAP1, so you have enhancement of NRF2, they keep this characteristic active for longer. And of course, when people moved to mice, they could show that when you delete NRF2, you have shortening of lifespan under ad libitum conditions. And when you activate it, you also attenuate different health span characteristics. During aging, we have hearing loss, we have changes in salivary glands. If you have activation of NRF2, you have attenuation of this decrease in these characteristics. But what was more striking in the rodents are the studies we have from long-lived naked mole rats. These are rodents that reach the age of 30 or 35 years. And in elegant studies, it was shown that their NRF2 is activated. You have higher NRF2 ARE binding activity. It was also shown that you have higher expression of NRF2, lower expression of KEAP1, and of course you have higher expression of all the target genes of NRF2. And when they tried in different long-lived rodents to see how this is performed, they could see that it's not mainly the higher expression of NRF2, but it is rather due to the reduced expression of the negative regulators. So KEAP1 and beta-TRCP are downregulated in these long-lived species, and this is how you have the activation of the NRF2 pathway. So the general consensus in the long-lived animals is that more or less we have higher levels of NRF2, lower levels of KEAP1. For sure, we have NRF2 nuclear binding activity that is high, and we have increased transcription of the downstream targets. And of course, there are also people that have tested when you have the negative effects of NRF2 hyper activation, and they suggested that maybe in these long-lived animals, maybe they have a broader range of set points before NRF2 levels result in negative results of NRF2 activation. And when we move to humans, there is a progeria syndrome called Hutchinson-Gilford progeria syndrome, where people have accelerated aging. They die maximum at the age of 20 to 25 years. They got, this is due to a truncated form of lamin A, which is called progerin. So there was a very nice study getting fibroblasts from patients with progeria, and they could show that progerin binds NRF2, and therefore NRF2 cannot access the ARE elements. Therefore, you have reduced transcription of the target genes, you have higher reactive oxygen species levels, and this is how you have the different phenotypes in these people. However, if you reactivate NRF2, they were able to show that they reversed the progerin-induced aging defects, and especially the defects that have to do with the oxidative stress and the reactive oxygen species. And there are a few studies talking about centenarians. Of course, more work is needed to be done, where it was suggested that the dietary habits of these centenarians the diet of those people is enriched in phytochemical and NRF2 inducers. And probably one of the reasons these people have reached to this high age is because they have, let's say, a transient NRF2 activation through their diet. And it was also shown that in these people, you have age-related trajectories in their human gut microbiota. And it was found that centenarians have a high antioxidant activity of gut-resident lactobacillus. But there is a surprise, lactobacillus species have been shown to induce NRF2. So it might also go through their microbiota, all this induction leading to this long age. So more or less, what is known nowadays for longevity is that in relation to NRF2, is that either through drugs and phytochemicals or through genetic activation of NRF2, you have increased detoxification, increased antioxidation, you have changes in the metabolism, anti-inflammatory conditions, and this is how you attenuate the age-related differences. So what is known from all different species that I have shown is that transient activation can be of KEAP1 NRF2 system can be beneficial for longevity, but persistent activation is not. So in my lab, we're working with a replicative senescence model, so human fibroblasts, that if you keep them in culture for 4 to 6 months, constant culture, they become aged, they become senescent, and this is the best model so far to recapitulate human aging. And I'm working also with the nematode worm C. elegans. Most of the pathways that are known nowadays to affect human aging, they were firstly identified in C. elegans and then verified in higher eukaryotes and then in humans. So these pathways are very conserved, and that's why C. elegans is a great model for aging studies. And I'm also working with the proteasome. This is the 20S core proteasome. It is a barrel-shaped multicatalytic enzyme composed by 4 different rings. The outer rings are made by alpha subunits. The inner rings are made by beta subunits. The alpha subunits are the binding sites for regulatory particles to be bound on the 20S proteasome and to give rise to supracomplexes of the proteasome. And they also form a gated channel through which the protein to be reintegrated will enter in order to reach the beta catalytic subunits. There are 3, beta 1, beta 2, and beta 5. They cleave after acidic amino acids, basic amino acids, and hydrophobic ones. And this is how you have the activities of the proteasome. Of course, you have different complexes of the proteasome, like the 26S, the 30S. It doesn't matter. It takes part in the normal, in the proteolysis of normal damaged and misfolded proteins and peptides for enzyme presentation. And therefore, it is not surprising that if you kill the proteasome, within 24 hours, you have massive cell death. And of course, it is also implicated in age-related diseases, in proteinopathies. You name the disease, Huntington's, Alzheimer's, Parkinson, polyglutamine disease, the proteasome is downregulated. And why do I talk about the proteasome? Before that, the proteasome, we have shown that the proteasome is downregulated during replicative senescence. If you partially inhibit the proteasome only by 50% in young cells, so you have young cells with the activity of an old cell, you have a p53-dependent premature senescence that is irreversible. And the older the donor gets, the lower the function and the activity of the proteasome. But if you go to the centenarians, they have an active proteasome. And we know that NRF2, many of the target genes of NRF2 are the 20S proteasome subunits. So this is how we came in contact with NRF2. So we had decided that we would go for compounds that are NRF2 activators. And through this activation, we were aiming to find activators of the proteasome. And we came up with 18-alpha-glycyrrhetinic acid. This is a triterpenoid. You can find it in Glycyrrhiza radix. And in the presence of 18-alpha-glycyrrhetinic acid, you have translocation of the newly synthesized NRF2 in the nucleus, and you have activation of the proteasome, which is totally NRF2 dependent. Because if you treat the cells with 18-alpha-glycyrrhetinic acid, but you silence NRF2 or you have mutated NRF2, you don't see this activation anymore. So we found the compound that activates NRF2 and the proteasome. And when we treat the cells throughout the lifespan with this compound, we have lifespan extension. And the cells that are treated with 18-alpha-glycyrrhetinic acid, they maintain their young phenotype for longer. When we moved to the organismal level and we gave 18-alpha-glycyrrhetinic acid in C. elegans, we could show again that we have, with different ways, that we have activation of the proteasome. And if we treat NRF2 with 18-alpha-glycyrrhetinic acid, the wild-type animals throughout their lifespan, we have change in the median lifespan, but also the maximum lifespan. And just to see whether it goes through NRF2, we used mutants for skin1. And when you treat the animals that are mutated for skin1 with 18-alpha-glycyrrhetinic acid, you lose the lifespan extension, thus showing that you need this activation of the proteasome through NRF2 to have the lifespan extension. And we also found that this activation of skin1 goes through phosphorylation by PMK1, and this is how you have the activation of skin1. We also used animals that are models for Alzheimer's disease. The nematodes do not have A-beta, but there are transgenic animals overexpressing human A-beta peptide in their body wall muscle cells. And the human A-beta peptide does what it does in our cells, so it gets aggregated. These aggregates are on the muscle cells of the animal, and eventually the animal gets paralyzed. So it's a pretty straightforward assay. And you can clearly see in different strains that if you pretreat the animals with 18-alpha-glycyrrhetinic acid and you activate skin1, then you have a decelerated paralysis. You see the red line that is shifted to the right that shows that they get paralyzed eventually, but much later as compared to the control animals. And if you get animals that overexpress human A-beta peptide linked to GFP, you can practically see in vivo in the animals the aggregates, and you can see how less aggregates you have in the presence of the NRF2 inducer as opposed to what you have in your control animals. And we also used murine cortical neurons, and we treated them exogenously with human A-beta peptide. Normally they die. This is a huge shock for them. But we also pre-treated the neurons with 18-alpha-glycyrrhetinic acid, and we allowed the activation of NRF2 and the proteasome. and we could see lower cell death, thus showing the protective effect of this NRF2 activation. We have also created genetically overexpressing the proteasome animals. So we chose the PBS5. This is the beta-5 catalytic subunit of the proteasome in C. elegans. And now by overexpressing this subunit, we could see lifespan extension. And then we were wondering which transcription factors play a role in this lifespan extension. And of course, we came again up with skin1. If you kill skin1 in the PBS5 overexpressing animals, you don't have any more this lifespan extension. That's showing that is really, really important. And since in aging, you have also huge DNA damage, we were wondering whether NRF2 can also play a role in DNA damage and protection against DNA damage. And we came up with the following experiments. So if you treat normal cells, primary cells, with mitomycin C, this is a cross-linking agent that causes DNA double-strand breaks. You have differences in DNA topology, and you have increased also reactive oxygen species production. And of course, you have high cell death as compared to what is happening to the control cells. Nevertheless, if you add mitomycin C, but also 18-alpha glycyrrhetinic acid, and you allow the NRF2 activation, you can clearly see less cell death, thus showing a protective effect through this NRF2 dependent activation. If you see here the foci, the gamma H2AX foci that are formed when you have DNA damage in the cell, here, when you treat the cells with mitomycin C, you can see that almost all cells are full with these foci. However, if you treat the cells also with 18-alpha glycyrrhetinic acid and you allow NRF2 to act protectively, you can clearly see how you have decreased foci. And here with the COMETS assay, you can understand that you have less DNA damage when you have the activation of NRF2. And just to see through which pathway you have this, we came up with ERK, NRF2 signaling pathway. And just focus here. Here you have cells that are treated with mitomycin C, and you have these foci. However, here you have cells that are treated with mitomycin C, but on top of that, you also have, they are treated with 18-alpha glycyrrhetinic acid, but they are also treated with this inhibitor of this ERK NRF2 pathway. And you can see that you don't have this protective effect anymore. That's showing that this protection comes from the activation of NRF2. So in conclusions, NRF2 may represent a master regulator of aging, longevity, and health span, as it has been already suggested. But you have to be sure about the exact conditions under which these beneficial effects occur. Timing and dosage is very important. And you have also to know and to be sure and to identify the side effects, including the effects of the potential NRF2 activation in pre-malignant cells that can occur in the body and we are not aware of. So with that, I would like to thank you for your attention. I haven't mentioned the work from Anna and Lena that were pivotal in the lab, but still I have to mention them. And I have to mention Ben Bedfar and Antonio, thank you for everything. And I leave you with that. At the end, it's not the years in your life that count, but it's the life in your years that count. So it's okay if we don't extend the lifespan as soon as we can extend the health span. And you are all invited in the Society for Free Radical Research annual meeting this June in 2026 in Mainz, Germany. Thank you very much.
And thank you, Nikki, for your interesting talk. Now we can move to the second speaker. And I'm pleased to introduce Dr. Alessandra Berri, a researcher at Istituto Superiore di Sanità in Rome, Italy. Her research explores how psychophysical and metabolic stressors interact to influence mental health across the lifespan, from prenatal development to aging, with a particular focus on oxidative stress as a shared underlying mechanism. The title of her talk is NRF2, unbalanced nutrition and vulnerability to chronic non-communicable diseases from early development to senescence. I am pleased to invite Dr. Berry to take the floor.
Thank you, Brigitta. I'm gonna share now my presentation. Can you all hear me?
Yes.
And see the presentation? Okay. So as Brigitta just told you, I'm Alessandra Berry and I work...
Alessandra, I don't see your presentation. Try again to share your screen.
I will try again. Can you see it now?
Yes, now see, yeah. Put in full screen mode, okay, perfect.
Okay, perfect. Okay. Okay, so as Brigitta just told you, I'm Alessandra Berry and I work at the Istituto Superiore di Sanità at the Center for Behavioral Science and Mental Health. And today I would like to present some very interesting results from our research project that have as a common ground the characterization of the negative effect of obesogenic diet throughout life. And we specifically focused on oxidative stress as a main mediator of these negative effects, paying attention also to the role played by NRF2. We are all facing during the last decades the consequences of what has been defined the nutrition transition, a condition characterized by the increased amount of the consumption of hypercaloric food, poor in nutrients, and a decrease in physical activity. And this nutrition transition has had as a main consequence the global growth in the prevalence of chronic non-communicable diseases. And I'm not only referring to metabolic disorders like cardiovascular disease or type 2 diabetes or fatty liver, but also I'm referring also to neurodegenerative disorder and psychiatric disorders. A common feature characterizing these chronic diseases is the increase in inflammation. However, growing evidence during the year is showing that oxidative stress may also play a pivotal role participating in fundamental signaling cascade, dealing with neuroplasticity and also neurotoxicity. Now, the effect of a metabolic challenge can vary a lot based on the lifetime during which it is experienced. For instance, during fetal development, an obesogenic diet can lead to embedded biological traces leading to long-term health outcomes throughout life. By contrast, if experienced during senescence, a main consequence is to accelerate the aging process, leading not only in a decrease in lifespan, but also in health span, as brilliantly introduced before me, Nikki. Now, pregnancy may surely represent a sensitive time window in the life of a mother, since the mother has to adapt her metabolic balance to meet the requirements of the growing fetus. However, it represents also a key time during the life of the developing fetus. In fact, the environment during this time plays a pivotal role by interacting continuously with the genetic background in shaping the developmental trajectories to prepare the growing organism to face the life after womb. Thus, an obesogenic diet experienced during this time may powerfully increase maternal inflammation and oxidative stress, but also may increase the amount of glucocorticoids, which are the main stress hormones. And these hormones have the power to decrease the shield provided by the placenta, exposing the fetus to an unwanted amount of altered maternal hormones and cytokine and metabolic mediators. Now, during the year, we have gained substantial experience in characterizing the negative effect on the offspring throughout life of a maternal high-fat diet. And we achieved this great experience by means of animal models, paying attention also on sex differences. We made use of very standardized animal model, and briefly what we did was to start feeding with an obesogenic diet female breeders, and after 10 weeks on the diet, we start also the administration of N-acetylcysteine, which is a powerful antioxidant. Then we went ahead with the administration of both the diet and this antioxidant throughout breeding and until birth. After that, all the pups were went onto a standard diet. And in this experiment, I will briefly show you, we focused on the effect of this metabolic stressor during pregnancy, specifically in adolescent offspring. We selected this time because we know that in humans during adolescence, it can be easily observed the first onset of psychiatric disorders. So at 1 month of age, we went ahead with the characterization of the metabolic and behavioral phenotype of male and female pups, and then we sacrificed the animals to collect brain tissues and blood samples. When we looked at the emotional profile, we made use of the elevated plasmates test. For those of you who are not familiar, this is a test run on this apparatus, that is a cross-shaped apparatus elevated from the floor by 40 centimeters that has 2 enclosed arms and 2 opposite open arms. And usually the more the time spent in the open arms, the more the explorative and disinhibited behavior of the animals, while the opposite held true when the animals spend more time in the closed arms of the mates. As you can see here, we could nicely observe the effect of the maternal obesogenic diet, specifically in females. So it was a gender-specific effect. And high-fat diet increased the amount of time spent by female mice in the open arms of the mates. This effect was nicely paralleled by the levels of NRF2 and total BDNF. Basically, we focused on the hippocampus, which is a brain area in the limbic system involved in the modulation of cognitive abilities and also of emotional behavior. And we could nicely observe that the levels of NRF2 were specifically decreased in female subjects upon high-fat diet, maternal high-fat diets. When looking at the BDNF, this effect was even more interesting. In fact, we could still observe a decrease in female subjects of total BDNF level upon high-fat diet, but this effect was nicely reverted by the administration of N-acetylcysteine. And BDNF, for those of you who are not familiar, is a neurotrophin that plays a key role in neuronal differentiation during development, but also in brain plasticity, cognitive abilities, and emotional behavior. Now, these effects were maintained over time. In fact, in different batches of animals using exactly the same protocol, but tested at adulthood, we could still observe this disinhibited behavior specifically in female subjects in the elevated plasmates test that was nicely prevented by the administration of N-acetylcysteine. Once again, when looking at the brain, NRF2 level were decreased in female subjects, but the effect was counteracted by the administration of N-acetylcysteine. And the same held true for BDNF. Nicely, we could also observe an effect on glutamate cysteine ligase, which is the rate-limiting enzyme for glutathione. Again, high-fat diet, this maternal metabolic stress was able to decrease the level of this rate-limiting enzyme specifically in the brain of females, but N-acetylcysteine was able to prevent such a negative effect. Looking at the different brain area, however, like the hypothalamus, which is a brain area mostly involved in the mediation of the effect on metabolism, it mostly regulates metabolism. The effect, the gender effect shift towards male. In fact, The effect of the obesogenic diet was observed in adult males upon high-fat diet and was only in adult males and was nicely prevented again by N-acetylcysteine. Now, keeping in mind these sex differences, a nice question, and going back to my previous slide, what happened if such a metabolic stress is experienced in a different time during life, such as during senescence? Senescence, in fact, is another critical time during the life of an individual characterized by a decrease in brain plasticity and also characterized by a progressive loss of homeostatic control and represents per se a risk factor for the onset of metabolic and mental health issues. And this progressive loss to maintain homeostasis relies upon the continuous interaction between peripheral and central tissues. And as far as the mechanism are concerned, we can observe under physiological condition an increase in oxidative stress, low-grade chronic inflammation, and a massive hormonal remodeling. Now, the answer to this question is quite easy. If a metabolic stressor such as westernized style diet is experienced during senescence, we can observe the shift from what is a physiological aging to a pathological aging that might affect not only lifespan, but also, and most importantly, health span, by increasing much more the risk to develop dementia and also conditions such as metabolic syndrome. The interest of current gerontoscience is progressively shifting to investigate the power of a natural compound to promote health span. And this is because of several reasons. First of all, most natural compounds share antioxidant and anti-inflammatory properties. They've been shown to be metabolic modulator, they are neuroprotective, and most interestingly, their action is pleiotropic with reduced side effect compared to most target-specific drugs. Again, we took advantage of animal models to test the ability of one specific natural compound, which is rosmarinic acid, a polyphenol that is found in several herbal plants, to prevent the negative effect of metabolic challenge, such as westernized diet. For those of you who are not familiar, the average lifespan of mouse breeders is 24 months. So a mouse when is 20 month old is already quite old. So we started administration of rosmarinic acid in drinking water in 21-month-old male and female C57 mice. And then after 2 weeks on the rosmarinic acid, we started administration of this extreme metabolic challenge. After 4 weeks, we started characterization of the behavioral and metabolic phenotype of the subjects, and then we sacrificed the animals. And once again, we collected the hippocampus. This time, we preferred an untargeted approach to specifically observe the effects both of the rosmarinic acid, but also of the metabolic challenge in eventually leading to cognitive disabilities and the ability of rosmarinic acid to prevent such effects. We took advantage of this very popular test for assessing cognitive abilities, which is the Morris Water-Mates test. This is a swimming pool filled with water at room temperature, and there is a platform hidden below the water surface. And looking at the time spent by the mice to reach the platform, the latency to reach the platform is a good indicator of cognitive abilities. So when looking at male and female subjects, we could observe clearly the negative effect of the obesogenic diet since it increased the latency to reach the platform. However, rosmarinic acid was able to prevent such an effect, both in male and female mice. Then we looked at insulin resistance by means of the HOMA index, and we adapted this measure of insulin resistance for mice. And we could nicely observe here a sex-specific effect, as only male mice show an increase in insulin resistance. But again, in this case, it was prevented by rosmarinic acid. As far as our untargeted approach to transcriptomic in the hippocampus is concerned, we can nicely see here again a sex-specific effect. In fact, looking at the brain of female mice, we could observe overall an upregulation of most pathways, both as a result of rosmarinic acid as well as Western diet administration. Looking at male subject, the opposite held true. And most intriguingly, the most great effects were observed upon Western diet. Looking at the specific pathways that were modulated, some of the specific pathways that were modulated as a result of rosmarinic acid or Western diet, while in females, most pathways were related to inflammation and also brain plasticity. Looking at male mice, we could observe a nice regulation of glutathione metabolism. And looking at the effects of Western diet, most of the pathways were related to changes in metabolism, in pathways dealing basically with metabolism and cardiovascular disease. Then we try to characterize another very interesting compound that is trehalose, but this time we focused on natural aging, on physiological aging. And we started administration to 25-month-old male and female mice. So these mice were really, really old. And after a basal assessment of their motor function, we started administration with trehalose. So this time, no challenge, no metabolic challenge. After 4 weeks, we characterized the motor function of these animals and also motor learning abilities. And when looking at results, again, we could see very nice sex-related effects. First of all, when looking at body weight, that is a very important indicator of health, especially in very, very old mice. We could observe an effect specifically in females that reached 26 months of age. This effect was nicely prevented by the administration of trehalose in drinking water. Looking instead at the Rotarod test, which is a motor test that relies on the ability of mice to learn motor coordination. So there is a rotating rod, and from trial to trial, the rod accelerate, so increase the velocity, and mice have to learn to move and to coordinate their forepose in order not to fall from the Rotarod. We could observe an effect on control mice. Control mice at the third trial show reduced latency to fall from the rod, while this effect was nicely prevented by the administration of trehalose. Looking at the frontal cortex this time, that is most involved in motor learning, the effect was nicely mirrored by the increase in NRF2 protein, specifically in males. And this effect was also strengthened when looking at the autophagy, again in the frontal cortex, specifically of male subjects. So, to conclude, I hope I convinced you that obesogenic diets represent a powerful metabolic stressor, whether administered during development or during senescence, and affect not only brain function, but also behavior and metabolism. Oxidative stress seems to be a main mechanism mediating the sex-dependent effects of such stressor, and the modulation of NRF2 is probably underlying these sex-specific effects. NRF2 is finely tuned both by synthetic and natural compounds that hold antioxidant properties. and should be considered as a target of interest for pharmacological strategies aimed at containing or counteracting the effect of obesogenic diet on emotionality and cognitive abilities. I would like to thank all the collaborators at Istituto Superiore di Sanità, at University of Milan and Sapienza University and University of Zurich. Thank you for your attention.
And thank you, Alessandra, for your interesting and informative talk. So now we are in time and we can move to the other speakers. It's my pleasure to introduce Dr. Andrea Neves Carvalho, Assistant Professor at the Faculty of Pharmacy, University of Lisbon. Her research focused on the molecular mechanism behind the mitochondrial dysfunction, oxidative stress, endoplasmic reticulum stress, how these processes contribute to neurodegeneration, aiming to identify novel therapeutic targets for neurodegenerative diseases. The title of her talk is NRF2 Crosstalk with the Unfolded Protein Response in Parkinson's Diseases, Role in Redox homeostasis and proteostasis restoration. Please join me in welcoming Dr. Carvalho.
Thank you so much for the kind words. And thank you, Brigitte, for inviting me and the organizers for allowing me to share my work with you. So, can you see already my presentation?
Not again. Try again. Okay, yes. Put the screen in full mode. Okay, perfect. You can
Okay.
go.
Yes. Is everything okay?
Yes, yes, you can go.
Yes. Okay. Thank you. So today I will talk to you about, of course, NRF2, which is our common interest, but also the unfolded protein response in Parkinson's disease. So first of all, just a very brief introduction for those who are not in the field. Basically, neurodegenerative diseases are a group of very heterogeneous age-related disorders. So the main risk factor for neurodegenerative diseases is aging. And these diseases are typically characterized by progressive loss of specific populations of neurons, leading to loss of structure and function in certain regions of the brain. That then affects structure and function. And despite displaying different pathological features and affecting these different regions, these diseases share common hallmarks and common clinical features. Some of them are related to misfolded protein accumulation and, of course, loss of neurons. And a common factor that is throughout the development and the progression of these diseases are the stress responses. So the activation of different stress responses, oxidative stress and the unfolded protein response, and also other stress responses. So Parkinson's disease is a neurodegenerative movement disorder, which is characterized by selective and progressive loss of dopaminergic neurons in the substantia nigra, pars compacta, and the accumulation of intercytoplasmatic inclusions known as Lewy bodies. Predominant PD motor symptoms arise from the loss of dopamine in this region. And despite both environmental and genetic factors have been related with the disease, its cause is still not completely clear. And a lot of factors have been described to be involved in the progression of the disease. So solid evidence has accumulated for the involvement of neuroinflammation, impaired autophagy and proteasome pathways, mitochondrial dysfunction, and accumulation of aggregated proteins. So NRF2 doesn't need any presentation, but still, I'm just going to do a brief presentation here because I think it's worth. So the NRF2 is one of the primary oxidative stress sensors in the cells, and accordingly to the most commonly accepted model for the regulation of its activity. Under basal conditions, NRF2 is sequestered in the cytoplasm by the complex with KEAP1 and the E3 ubiquitin ligase complex that targets NRF2 for degradation for the proteasome. By this way, KEAP1 represses NRF2 transactivation activity and accelerates its degradation to the proteasome. But under conditions of oxidative stress, there is a disruption of this complex and stabilization and accumulation of NRF2 in the cytoplasm, which allows that newly synthesized NRF2 be translocated to the nucleus where it heterodimerizes with other factors and leads to the transcription of several cytoprotective genes. So one of these genes that I would like to highlight, because NRF2 is involved in the transcription of many cytoprotective genes, more than 250, involved in different and important protective pathways, but some of them are xenobiotic metabolism. And I'm particularly interested for many years in this protein that is glutathione S-transferase B. And why did we decide to study GSTP in the context of Parkinson's disease? Because GSTP is a phase 2 drug metabolizing enzyme that its main function is to catalyze xenobiotic biotransformation and detoxification. But this gene is highly polymorphic, and it confers higher polymorphisms in this gene confer higher susceptibility to sporadic Parkinson's disease. Moreover, glutathione S-transferase B has been shown to protect cells from reactive oxygen species through its catalytic activity of conjugation with reactive metabolites with glutathione, but also through the alteration of glutathione levels, and as I'm going to show you, to the control of the catalytic activity of JNK and modulation of protein S-glutathione. So to study GSTP, we used, in the context of Parkinson's disease, we used an experimental model of PD that consists in the systemic administration of a single dose of 40 milligrams per kilo of MPTP, which is a known neurotoxin that induces PD-like symptoms. And once in the brain, so the MPTP crosses the blood-brain barrier, and once in the brain, it's metabolized into its active toxic metabolites, MPP+, which then accumulates because it enters the neurons through the dopamine transporter and accumulates in the neurons, leading to an inhibition of the complex one of the respiratory chain, or blocking it and leading to the consequent production of reactive oxygen species and damage to the cell. Many years ago, it was shown that only after 3 hours of systemic injection, there's already a peak of these toxic metabolites in the brain. So first of all, we confirmed, validated in our model that the systemic administration of MPTP led to loss of dopaminergic neurons together with the generation of striatal fibers. And what we observed is that in knockout mice, for GSTP, this loss of neurons and loss of striatal, so this striatal fibers degeneration occurred at an earlier time point. So GSTP knockout mice seem to be more susceptible to neurotoxic effects of MPTP, displaying a significant loss of negral dopaminergic neurons at an earlier time point when compared to their wild-type counterparts. Given that one of the suggested main non-catalytic functions of GSTP is through the regulation of J and K, we next went to explore this stress-induced pathway. And what we observed was a transient increase in the levels in the levels and activity of J and K as early as 3 hours post MPTP administration. Interestingly, this increase was significantly higher in the knockout mice. And we further wanted to explore and characterize this J and K activation cascade. And for that, we assessed the activity towards c-Jun, which is its target of this kinase. And then we performed a co-immunoprecipitation assay to evaluate alterations in the protein-protein interaction between J and K and GSTP. And we showed for the first time in vivo that the direct interaction between J and K and GSTP, both in the striatum and midbrain of these mice. Another important role that GSTP can have is through modulation of glutathione, which so the versatility and reversibility of the cysteine tiols serves as basis for the specificity and the diversity of many redox signaling pathways. And mainly, this reversible redox switch operates through post-translational modifications of these cysteine groups. And glutathione transferase P is in fact relevant for the process of glutathionylation. So, taking into account that glutathione maintains the redox status of protein sulfhydryls through glutathionylation, a reversible post-translational modification that might temporarily protect these cysteine residues from irreversible oxidation, and that the key distinguishing feature of KIP1 is its higher number of cysteine residues, and that modification of critical cysteines in KIP1 through bioxidants and the electrophiles is one of the mechanisms that activates NRF2. We wanted to further explore the role of glutathionylation using these knockout mice. So, first of all, we went as an indicator of the changes in the cellular redox status induced by MPTP. We measured the levels of total free glutathione and both reduced and oxidized forms. And there is a significant decrease in both total free glutathione and reduced glutathione. And although the reduced glutathione, the amount of reduced glutathione decreases, there is not accompanied by an increase in the oxidized glutathione. Therefore, we also assessed some of the enzymes that are involved in the regulation of the GSH-GSSG equilibrium. And what we observed was an increase in the GPX activity and a decrease in the GR activity, which indicated that the recycling of glutathione was not being efficient. Therefore, and if we didn't see an accumulation of GSSG, we had the idea that it might be due to it being used in the glutathionylation process. And in fact, we evaluate the general glutathione by non-reducing Western blot and detect a different pattern of glutathione proteins in the wild type and GSTP knockout mice. Importantly, by co-immunoprecipitating, we demonstrated that MPTP-induced oxidative stress led to specific increase in the levels of S-glutathione KEAP1 in the brain of the wild-type mice, but that doesn't happen in the knockout mice. So it seems that GSTP is necessary for KEAP1 glutathione. The specific modification of KEAP1 cysteine residues also led to an increase in the translocation of NRF2 to the nucleus, and in accordance, we also observed increased levels of some of NRF2 targets. Furthermore, to explore in silico, to corroborate our data in silico, we used a crystallographic structure of KEAP of the Kelch domain from human KEAP complex to its NAH2 domain of NRF2, and we identified two cysteines that are potential glutathione sites. However, only cysteine 434 is at the surface of the protein close to the binding pocket, being more susceptible to be modified. And in fact, using different docking poses, we showed that when the NAH2 domain of NRF2 is docked inside the Kelch domain from the human non-glutathione KEAP1, it interacts with these residues. However, when we convalently dock GSH to cysteine 434, we saw that GSH also interacts with the same amino acids, partially occupying the pocket previously occupied by NRF2. So when to this glutathione Kelch domain we docked NAH2, we saw clear modification of the position inside the binding pocket, which indicates that the fact that that KEAP1 is glutathione alters its complex with its ability to complex with NRF2. So in these two works, we demonstrated in vivo that GSTP modulates the response of MPTP induced oxidative stress by acting as an endogenous regulator of JNK activity through protein-protein interactions, and that it promotes the glutathione of KEAP, an endogenous regulator of NRF2, leading to NRF2 activation. And this is a positive feedback regulatory loop that represents a novel mechanism through which GSTP might elicit antioxidant protection in the brain. So, more recently, and continuing with this, with our interest in the redox, in assessing redox proteins in the brain. We had access to human PD brain samples, and we performed a global redox proteomic analysis of PD patients' brain samples, focusing on redox post-translational modifications, and we used a method that enabled us to simultaneously examine the alterations in total protein levels between PD and control samples, but also alterations at the level of the redox proteome. And we had some proteins that were simultaneously altered in terms of their levels and in terms of their presences in the reduced fraction or oxidized fraction. Interestingly, this protein, aldehyde dehydrogenase 1A1, which is also an NRF2 target, was identified in both fractions, and it has been recently described to be a marker of vulnerability of dopaminergic neurons to degeneration in Parkinson's disease. So another hit that we got from this analysis was in terms of the unfolded protein response. And since neurodegenerative diseases are also considered protein misfolded disorders, and protein aggregates were detected in the tissue of patients with neurodegenerative diseases and associated with the presence of these unfolded protein response markers, we decided to further explore a bit this pathway. I have to be quick because I think I'm a bit up to time. But so basically, in response to ER stress, a very coordinated signal transduction pathway, the unfolded protein response, is initiated. The UPR triggers a series of adaptive signaling pathways destined to improve protein folding and degradation and to enhance protein quality control mechanisms. Basically, there are three main branches of the unfolded protein response that are activated, and they have specificities related with the context, disease context dependence, and also time dependence. And as I've said, they can activate both adaptive responses, but also pro-apoptotic responses in response to a more chronic stress. So in the samples of the human PD patients, we assessed the levels of some of these UPR markers in collaboration with Dr. Jeroen Huismans and Dr. Wiebe Schepper from the Netherlands, which have previously also studied the RCS in the PD brain. And we have verified that there is an increase in all three main branches of the UPR. However, when we further explored the pathways, one of them stood out, and it was the PERK branch of the UPR. We saw a tendency to an increase in the ATF4 levels and also in SHOP. So I will pass this one. So we got back to our model, and we explored in this MPTP mouse model. We tried to explore more this UPR pathway, and we saw an activation of this branch that you can appreciate here, which appears to be activating an apoptotic response. Therefore, and since we in our group we work for a long time with TUDCA, which is taraursodeoxycholic acid, which is an endogenous bile acid with several neuroprotective properties that we have previously shown that was protective in this same mouse model by improving motor symptoms. And also that this effect was partially mediated through an activation of NRF2, we now decided to explore if, since TUDCA is also described as an ER chaperone, we decided to see if it could have an effect in recovering from from ER stress and preventing ER stress. So as you can appreciate here in this preliminary data, the QDCA treatment appears to somehow reduce the activation of this branch of the UPR in this mouse model. And here, just to show you that we have a slightly different response in the knockout mice when compared to the wild-type mice. But this was not a surprise, since we have already observed that in basal levels, knockout mice that we have also previously shown that are more susceptible to the MPTP insults and that have activation of a lot of pathways, are also displaying an increased level of, for instance, of the SHOP gene. So we are now currently exploring more the mechanisms that might underlie this modulation of the UPR by QDCA in this model, and also the motive for a GSTP to be GSTP knockout mice to be more apparently more susceptible to this insult. So finally, I would just like to thank the people in my group and some collaborators, and thank you for your attention.
Okay, thank you very much, Andrea, for this nice talk. Everybody is welcome to add their comments and in the chat. And now we are going to talk. We are going to introduce our last speaker, who is Professor Fabio Di Domenico. Fabio is a professor at the Sapienza of Biochemistry at the Sapienza University of Rome, and he has been working mainly on issues related to oxidative stress, proteostasis in models of neurodegeneration, taking as a model the Down syndrome. And I think we are also honored to have here with us Professor Alan Butterfield, who is probably your mentor from your previous studies in the United States. So please, Fabio, go ahead.
Oh, thank you, Antonio, for the nice words, and thank you, Brigitta, for inviting me. Just share my slides. Hope you can see everything.
Yes, yes.
Okay, perfect. As I told you, thank you, and also thanks to Professor Butterfield to being here. Today, I will show you some of my recent data concerning the interplay between proteostasis, metabolic alteration, and how NRF2 can be involved in this interplay in the context of neurodegeneration, and in particular, as I will show you in Down syndrome. So first of all, as you know, proteostasis is very important for brain health, and any alteration on the mechanism that govern proteostasis can lead to the accumulation of unfolded and misfolded proteins, and this can lead to neuronal loss, degeneration, can lead to loss of brain signaling, and also to neuroinflammation. And this might culminate into the development of the so-called protein deposition disorder, which includes also, as you know, Alzheimer's disease, Parkinson's disease, as we heard before, and also Huntington disease, among others. In the last decade, we have been involved in the study of Down syndrome neuropathology, since also Down syndrome share many characteristics of the protein deposition disorders. As you know, Down syndrome is the most common genetic cause of intellectual disability and is characterized by the trisomy of chromosome 21. Down syndrome affects multiple organs, including the brain, and individuals with Down syndrome develop an early form of Alzheimer's disease. Epidemiological studies demonstrate that the development of Alzheimer's disease comes very early in Down syndrome individuals at about 40 years old, about 2 or 3 decades before what happens in general population. And at about 80 years old, 60 years old, 80% of Down syndrome individuals demonstrate the development of Alzheimer's disease. Okay. So the study of autoptic brain from individual with Down syndrome demonstrates the accumulation of senile plaques, which are formed, as you know, by the accumulation of beta-amyloid, which is already present in the brain from Down syndrome individual at about 15 years old. Is also present the formation of neurofibrillary tangles composed of hyperphosphorylated tau that is also present very early, already in fetal brain can be detected, but also other characteristic features such as synapse loss, decreased brain size, and the increase of oxidative stress. The reason behind the early development of these neuropathological features need to be searched by mapping chromosome 21, and several genes present on chromosome 21 and also their protein products are directly involved in the neuropathological mechanism. For example, we know that APP, the amyloid precursor protein, when is overexpressed, leads to the formation of increased A-beta monomers, oligomers, and fibrils. DYRK1A is a kinase that is also involved in tau hyperphosphorylation, and so lead to the formation of tau tangles, but also the overexpression of SOD1 that is also present on chromosome 21 leads to the increased formation of hydrogen peroxide, which is not buffered by the induction of catalase. Another important gene that is present on chromosome 21 is BAC1, that many of you might know that is a repressor of transcription that competes with NRF2 in the induction of the antioxidant response. What we found in our initial study is an overexpression, of course, of BAC1 in the brain. This is the frontal cortex of a young Down syndrome individual and Down syndrome individual with Alzheimer's disease compared with age-matched control. As well, we found also the overexpression of BAC1 in a mouse model of Down syndrome at TS65DN at 3 and 15 months of age compared with age-matched control, meaning that this is a stable overexpression of BAC1. The overexpression of BAC1 was also associated with the increase of oxidative stress that we found also in Down syndrome brain and also in mouse model of Down syndrome. So given this pro-oxidant environment that we found in Down syndrome, we decided initially to apply a redox proteomics analysis to find a protein that can be oxidatively modified by the increased oxidative stress, proteins that are not working properly and can also alter the pathway in which they are comprised. We found that several proteins that are oxidatively modified are component of the proteostasis network, and in particular component of the unfolded protein response of the autophagy lysosome pathway, but also of the ubiquitin protein system. So this led us to think that we have reduced degradation, and we started to go more in depth in the analysis of those pathways. We started initially with the ubiquitin protein pathway, and we found, for example, that in Down syndrome brain, again, in young Down syndrome and Down syndrome with AD, there is an increased polyubiquitination, and this was associated with a reduction of the activity of the proteasome, measured as far as chymotrypsin-like, trypsin-like, caspase-like activity. So the proteasome, in this case, was not working properly. Then we follow our analysis on the proteostasis network, analyzing the autophagy lysosome pathway. I show you here one marker of autophagy, that is LC3-1, which is a marker of autophagosome formation. And we found that the reduction of autophagy was also associated with an increase in phosphorylation, and so of activation of mTOR, which is a master regulator of autophagy that negatively regulates the induction of the pathway. This was altered in Down syndrome brain, in the brain from individual with Down syndrome, but also in mouse model of the disease. Subsequently, we also used, for example, rapamycin to try to restore the pathway, but rapamycin is an inhibitor of mTOR, and we showed that was able to rescue mTOR and then autophagy, and also to reduce oxidative stress. However, what we focus very recently, and this is something that was also mentioned before by Andrea, is the analysis of the UPR, the unfolded protein response. So as you heard before, the unfolded protein response is comprised by 3 different arms or pathway, which are ATF6, PERK, and IR1A. We analyzed initially their activation in Down syndrome, and in this case, in Down syndrome mice, again, is the TS65DN mouse model. We found that among the 3 pathways, the one that was hyperactive in Down syndrome was only the PERK pathway. Moreover, several studies in Alzheimer's disease also demonstrated an hyperactivation of the PERK pathway that was detrimental in this case and was associated also with increased phosphorylation of tau, increased accumulation of Aβ. So we decided to focus on the PERK pathway. The PERK pathway, as you know, is activated by the phosphorylation of PERK, and upon phosphorylation, PERK then is a kinase that can phosphorylate initially eIF2 alpha, which upon phosphorylation also regulate protein translation, is a repressor of translation, but also activate ATF4, which control, induce several pro-survival pathway. Moreover, PERK can also regulate NRF2 because can phosphorylate NRF2 on serine 40, and upon phosphorylation, NRF2 can translocate into the nucleus and induce the antioxidant response. This is what happens normally in physiological stimulation. However, when we have a chronic toxic insult, the PERK pathway can become detrimental, can lose its regulatory capability, and can promote the pro-apoptotic signal. So what we initially did was to study the induction of the PERK pathway in our brain sample from young Down syndrome individual, in this case is always frontal cortex, and Down syndrome individuals with AD. What we found is that we have an hyperactivation of the PERK pathway. As you can see, we have hyperphosphorylation of PERK, hyperphosphorylation of IF2 alpha in both young and Down syndrome individual with AD. However, what we also found is that there was an uncoupling with the NRF2 signal, because we found a reduction of NRF2 protein levels, also reduction of NRF2 phosphorylation. Of course, this was mostly associated with the overexpression of BAC1, which, as I show you, is always increased in Down syndrome brain. To further go deep in the mechanism that govern this uncoupling, we also analyze the brain, in this case is IPL, from 3 different stages of Alzheimer's disease: preclinical Alzheimer's disease, mild cognitive impairment, and late stage Alzheimer's disease. What we found is that in the last 2 stages, so MCI and AD, an hyperactivation of the PERK pathway, you see PERK and IF2 alpha, this was similar to what we observed in Down syndrome. Also, in those 2 stages, we have also reduction of the phosphorylation of NRF2, and this reduction was independent of BAC1 overexpression that you see doesn't change. So in this case, we think that the pathway is more associated, the alteration of the pathway is more associated with aging than with genotype, as instead we see in Down syndrome individuals. Then we decide to try to modulate this pathway. So we went back to our mouse model. We use in this case another mouse model that is very similar to the TS65DN, which is the TS2CJ mouse. We gave a treatment for 5 days by intranasal delivery of an inhibitor of PERK. The inhibitor is known as GSK414 and is an inhibitor of PERK phosphorylation. So we wanted to achieve a reduction of PERK activation to see if we can rescue the detrimental effect of PERK and UPR activation. What we found is that indeed PERK was reduced in phosphorylation with the inhibitor. Also, IF2 alpha was reduced in phosphorylation by using the inhibitor, you see here in gray. And also we have an improvement of protein translation, meaning that we have reduced the impact of IF2 alpha phosphorylation in the repression of translation. This is using the poromizing assay. Then we analyze oxidative stress and oxidative damage in particular, and we see that we have a reduction of 3T levels and HNE protein adduct when we have the inhibition of PERK. So we have a rescue from the pro-oxidant environment. What about NRF2? I remind you that we have normally physiologically a direct relationship between PERK and NRF2, so PERK can phosphorylate NRF2, but in this case we have a reduction of PERK phosphorylation with the inhibitor. So we didn't expect a change of NRF2, while instead we found an increased phosphorylation of NRF2 when we use the inhibitor. And this was also associated with an increase of translocation of NRF2 into the nucleus. Moreover, we found increased expression of NQO1, increased expression of HO1, and also a reduced expression of BAC1 that we think was mostly due to increased degradation. So this was surprising for us, and since it was not PERK that is phosphorylating NRF2, we went to search also which other kinases can be involved in this hyperphosphorylation of NRF2, and we searched for different kinases that are well known to phosphorylate NRF2. We measured several of them, and we found that the only one that was stimulated by our treatment was PI3K, which is hyperphosphorylated and potentially can increase the phosphorylation of NRF2. What was also interesting is that PI3K is an integral component of the insulin signaling, meaning that we might also stimulate metabolically with our treatment the insulin signaling. So at this point, given the fact that we were intrigued by our results, we try to understand if there might be also a connection between the modulation of the unfolded protein response and of the PERK pathway. In this case, the alteration of the insulin pathway, so a metabolic pathway that can change upon different stimuli, and also the induction of stress responses. In concomitance with our studies, we also performed studies measuring the brain insulin resistance in Down syndrome mice, and we found that also the induction of insulin resistance is one of the earliest mechanisms occurring in Down syndrome, and several different studies also demonstrated that there might be a connection between alteration of the brain insulin resistance of the insulin signaling, and also the development of brain insulin resistance and the alteration in the induction of the unfolded protein response. Moreover, to strengthen these ideas, we also ran very recently a proteomic analysis in young Down syndrome brain and Down syndrome brain with AD, in which we found again that the pathway overexpressed that are mostly alterated in those conditions involve energy metabolic pathway, but also the insulin signaling pathway of proteostasis, so you see mTOR and UPR, and also the NRF2 antioxidant response, meaning that those pathways are the ones that are initially altered that can also drive the neuropathological mechanism that we see with aging in these samples. So what we are currently doing is try to understand what might be the connection between metabolic alteration, insulin resistance, the induction of the PERK pathway, and so its overactivation, and also induction of stress response, and in particular of NRF2 response. We are using currently primary neurons and primary astrocytes from our Down syndrome model, the TS2CJ, and from euploids, In this case, we are using both neurons and astrocyte because, as you know, they rely on different metabolic pathways, since astrocyte is more glycolytic, while neurons relies mostly on OXPHOS for energy production. So they might have a different sensibility upon the induction of insulin resistance. Then we use a stressor to induce insulin resistance, which is the IPA treatment, so insulin and palmitic acid. We previously tested these stressors in other model, and we see that it was achieving increased insulin resistance. Then, upon the induction of insulin resistance, we were monitoring what was happening in the PER pathway. And to modulate the PER pathway, we use two different UPR modifying agents or PERK modifying agents. One is the GSK414 that we used before. The other one is ISRIB, which is not a direct inhibitor of PERK, but is an inhibitor of IF2 alpha phosphorylation, so it works downstreams of PERK. What we found is that by using IPA treatment, we were able to induce insulin resistance in our models in both neurons and astrocyte. As you see, we have a reduction of the activation of the insulin receptor substrate 1, and also a reduction of the induction of AKT, which is the downstream signals of IRS1. This reduction was similar in neurons and also in astrocytes. So we were able to induce insulin resistance in our model. We then analyze what's happening as far as the PER pathway. So we analyze initially the phosphorylation of IF2 alpha. We see that there is an increase of phosphorylation with IPA, meaning that we achieve a stress of the endoplasmic reticulum, and also we induce the activation of the PER pathway. This was reduced with GSK. We don't see much reduction of ISRIB, but then when we analyze protein translation with the puromycin assay, we see that there is a recovery of protein translation, meaning that the two inhibitors are working on restraining the inhibition of translation that we achieve with IPA. This was the result we obtained in neurons. These similar results, very similar, we obtained also in astrocytes, meaning that, again, we can induce in both models the induction of the PER pathway. We can recover this induction using the inhibitor of the PER pathway. What about at this point of NRF2? We went back on the analysis of BAC1 and NRF2. In this case, I show you the BAC1 NRF2 ratio in the gene expression. What we see is that we have an increase of the ratio, meaning that the BAC1 is overexpressed, and this can lead to a reduced induction of the pathway, as we see also by the protein expression of HO1. The similar effect was also observed in astrocyte, since we have, again, an overactivation of BAC1, reduction, an increase of the ratio between BAC1 and NRF2. This is smaller than what we observe in the neuron, but still is involved in the reduction of the activation of HO1. We then analyze also ROS production, and surprisingly, by using the DCF assay, we show that IPA induce an increase of ROS in the cell, both neurons and astrocyte, euploids, and Down syndrome cells. And this was reduced while we use our inhibitors, meaning that we have a rescue of oxidative stress, but this might be, at this case, independent of the activation of NRF2. To further confirm this data, we also used a different mechanism for monitoring oxidative stress. We use in this case a different assay, which is the Cell-Rox assay by cytofluorimetry. And again, we see that there is an increased production of ROS species with the IPA treatment, and then we have a reduction when we use our PERK pathway inhibitors. Again, we also monitor in astrocytes. In this case, only TS2-CJ astrocyte, we obtained similar results also in euploid. We performed a scratch assay to evaluate migration and invasion of astrocyte. What we see is that we have a damage of cell of astrocytes health with IPA. You see the scratch is not closed also after 48 hours, while using our inhibitors, we were able to improve the health of astrocyte and reduce the scratch, as you can see also from the graph. So meaning that our treatment was working, and what we are doing now is try to understand what other mechanism can be involved. And of course, we haven't seen an activation of NRF2, an induction of NRF2, but this might also encompass from different antioxidant response or the other antioxidant responses that devoid NRF2 activation. So we can conclude this talk saying that, of course, the interplay between proteostasis, metabolic changes, and redox responses is very complex. And we have seen that is influenced by many different factors. For example, the cell or the tissue or the organ we're using, the genotype of the model we are using, the pathological mechanism involved, but also the pathological stage that we are studying, but of course also age, sex, and of course also metabolic status. So when we study all these different mechanisms, those factors need to be always taken into consideration. Of course, we studied the DS neuropathology, and in DS neuropathology, this interplay is mostly modulated by genotype, which, as I told you, is pushing BAC1 to overexpression, but is also regulating aberrantly several other mechanisms that are involved in this interplay. And of course, also the alteration of metabolic changes that we have observed in early Down syndrome brain, it's playing a role in this delicate interplay. So my message is that every time, of course, that we think of targeting a different aspect of this interplay by pharmacological modulation or any different kind of modulation, we always need to address their interplay and their mechanisms that are regulating those interplays. So I would like to thank all the people of my lab, so Marzia Perluigi, Eugenio Barone, Antonella Tramutola, and Chiara Lanzillotta. Chiara did most of the work that you have seen on the PERK pathway. Of course, postdoc researcher and PhD student, and of course, Professor Alan Butterfield, that was my mentor and helped me to start my career at this lab. So thank you.
Okay, thank you, Fabio, but thank you to all our speakers for keeping perfectly on time. It made this session flow so well. We received some questions in the chat. Before we can start with the first question that is for Nikki Condroianni. Ok, I can read. Quindi from Tiziana Grassi, quindi your research, Nikki, you can, okay. Your research investigate one of the biggest questions in science. How can we stay healthier for longer as we age? What compounds are more interesting in delayed aging and protect us from age-related disease? What you can say?
Yeah, it's the question that everybody wants to find the answer. Anyway, from work in nematodes, in Drosophila, in different models, it has been shown that usually when we talk about natural compounds, okay, usually you have polyphenolic compounds, you have triterpenes, diterpenes, flavonoids, these kind of compounds. Almost all of them have antioxidant properties, but the work all over the world has shown that it's not enough. I mean, the biggest problem with the antioxidants is that, for example, vitamin E, is that we have never seen extension of lifespan when you have antioxidants and only antioxidants. So probably you need the antioxidant properties, sure, since we breathe, we have oxidative stress, so you need. But I think that you should also have, you know, what Alessandra said, the pleiotropic effects of different natural compounds. So to me, a good compound would be one with the antioxidant properties plus something like anti-inflammatory properties, anticellular senescence. This is very in at the moment. We are all looking for senolytics, so compounds that kill the senescent cells, compounds that help the mitochondrial function. So this is the kind of compounds that I think will work at the end. And actually, I think we are very lucky because these kind of compounds are in our diet. So I don't know whether I would go for a drug or I would go for a better diet, getting all these compounds from our diet that at the end have a positive effect. Regarding specific compounds, you can find quercetin, you can find 18-alpha-glycyrrhetinic acid that we found. We have found also hederagenin, sulforaphan, rosamarinic acid. You know, all these compounds have been shown to extend longevity.
No, thank you for your reply. So we can move from the antioxidant compound with the toward compound with the cytoprotective activity. Quindi not limited to their antioxidant ability, but for their cytoprotective activity.
So all these that I told you, usually they also increase stress resistance. So they are cytoprotective anyway. And there is a limit, and there is where NRF2 plays a role. I told you that the hyperactivation reduces lifespan. However, it extends stress resistance. And we have seen that after a point, you have a difference between the longevity and the stress resistance. So they are all cytoprotective. And depending on the dosage, you can have good effects or bad effects in longevity as well.
Okay, I can move to the other question from Professor Alan Butterfield. He writes, our lab showed the paralysis of elegans coincided with the presence of elevated protein carbonyl in C. elegans expressed in human AB42.
Yeah. Hi, Alan. It's very nice to see you again. Yeah, I know. Actually, this is how we thought that maybe by activating the proteasome, we can help against Alzheimer's, because, you know, when you have increased carbonylated proteins, the oxidized proteins, the proteasome there sure can play a role. And indeed, when we activate the proteasome, we downregulate the carbonylated proteins in C. elegans. So yeah, I agree.
Yeah. Great work, Nikki.
Thank you.
Okay, we can move on from Raul Castro, University of Padova. Question to Nikki Condroianni. Amazing talk, thank you. Can you speculate about the mechanisms of action by which you think 18GA activating NRF2 or skin1? How feasible would it be to develop a drug to enhance lifespan and health span extension based on?
Yeah, so I'm not speculating. We have found that 18-alpha-glycyrrhetinic acid activates ERK1 in human cells, and ERK1 phosphorylates NRF2, etc. In C. elegans, it was the P38 MAP kinase. It's called PMK1, and this is how 18-alpha-glycyrrhetinic acid activated skin1, again through phosphorylation. Regarding your last question, you know, okay, I would love to find a drug like that, but first of all, you cannot claim lifespan extending properties and sell something like that, one. And two, although I think NRF2 is really fascinating, I think it's also really dangerous, depending on the dosage and the timing, etc. So I don't know whether I would go. I think I would prefer to go with the diet solution. So, you know, you just add in your diet more vegetables or whatever have 18-alpha glycosylglyceric acid. And still there could be a problem because it has been shown that when you have too much 18-alpha glycosylglyceric acid, you change your blood pressure. So, you know, everything in aging is about equilibrium. So I think we are far from drugs, so, you know, with such a general mechanism like activating NRF2.
Okay, thank you, Nikki, for your reply. This question is from Alexandra, for Alexandra Berry. First of all, congratulations on your interesting presentation. Okay, considering your result with N-acetylcysteine, rosmarinic acid, triarose, which type of antioxidant or nutraceutica do you think hold the most translation potential for preventing early life or age-related metabolic and cognitive impairments? So,
okay, thank you for the question. And to relate to what Nikki have just said, one main important thing when thinking about natural compounds compared to drugs in general or target-specific drugs is not only their pleiotropic effects, but also their hormetic properties. That is quite interesting. And that means that these natural compounds hold the ability to prepare the organism to face further insults. And this is a very evolutionary conserved mechanism throughout different species. And this is also, I mean, it's important because I can think about the use of natural compound as a preventive strategies, something used like chronically and added to our usual diet. While I can think of more an acute action, for instance, with N-acetylcysteine. There are a lot of clinical trials also with N-acetylcysteine. N-acetylcysteine does not only hold antioxidant effects, but also many other effects, especially with regard to mental health. It is quite an intriguing compound. And what I would add is that for nutraceutical compound, I would not go for prenatal treatment because most of this natural compound might not be completely safe, might be either teratogenic or, I mean, you have to be sure that they are safe to administer during pregnancy. Well, for N-acetylcysteine, it is known that it's well tolerated during pregnancy.
okay, thank you, Alexandra. From Antonio Quadrado for you. Is there any reliable evidence showing a correlation between NFLD and cognitive impairment in humans?
Yeah, thank you, Antonio, for the question. Yes, there are a lot of observational data and also meta-analysis, and it is known, well, it is studied quite a lot, the brain-liver axis. And one of the main interesting is to characterize the NRF2 hemioxygenase 1 pathway. And Chinese medicine, for instance, is very interested in characterizing the effect of natural compounds to target these specific pathways. And it is known that curcumin is able to, for instance, to reduce damages to the liver and to improve cognitive abilities, especially in middle age, for instance, and in subject with non-alcoholic fatty livers, but also with metabolic syndrome.
Thank you, Alexandra.
Currently, we have a clinical experimentation on this, on curcumin, not on alcoholic fatty liver, but on cognitive disability in middle-aged with mild cognitive impairment and obesity, mild obesity.
Obesity is one of the risk factors for MASLD. So we can move on. Another question for Dr. Condroianni from Antonio Quadrato. Quindi Nikki, it's for you. Can we define a NRF2 activity test to determine the biochemical age? Another philosophical question.
Yeah, I guess you mean the biological age. Yeah, well, we could maybe find genes, target genes that are very specifically changing with age, because you need to have something that specifically changes with aging. And then if you have a higher biological age, this will not be changed so much. So I think maybe we could think about these kind of genes, but to be honest, I wouldn't go for NRF2 and the activity, because it's multi-parametric. It changes with diet, with stress, with everything. I think if I would have to bet on something, I would go for the levels of KEAP1 or TRCP beta. I think I would go for that. Because also, if you see in the long-lived species, this is what is changing. And then you have the differences in the activity of NRF2. So if I would go for a marker, I would go rather for this expression and not for, the activity of NRF2. My feeling.
Antonio, what is your opinion on this?
I think this is I was making this question because I have myself this issue that I really don't know how to deal with. But I think your answer is really very smart. I think it happens also when we look at the inflammatory signature, interleukins, everything is a mess because small changes already give very confusing results. So maybe with NRF2 we will come up with similar situations. And your idea of looking at KEAP1 and beta TRCP and maybe some other repressors or activators is really very interesting, and I want to think about it. Thank you.
Welcome.
And another question for you from Roberta Foresti. Why is the overactivation of NRF2 detrimental for aging, excluding effects in cancer?
Yeah, so this is also the paradox, because when you have you age, you have more oxidative stress. So everybody at the beginning was thinking that activation of NRF2 would be beneficial, but this is the paradox, that during aging NRF2 goes down. So for sure there is an equilibrium there. I can think about many pathways that change. For example, if you overactivate NRF2 and you have an acting antioxidant capacity, then you also kill the redox signaling. So, okay, too much oxidative stress is not good, but you need oxidative stress to have the redox signaling. So one could be that. In Drosophila, it has been shown that when you hyperactivate NRF2, you have changes in the mitochondria, you have changes in the mitochondrial complexes, you have lipid oxidation, you have changes in metabolic changes. So I think that I said that before, there is a very nice and elegant equilibrium. And if you change something, you might change something in a beneficial way, but other things might change in the other way. So I think that this is why we cannot have this hyperactivation. Probably we change also pathways, maybe pathways that we don't know yet as well, that at the end are toxic.
Oh, thank you for your reply, very exhaustive. Okay, we can move to a question for Dr. Carvalho. Andrea, you can unmute your microphone. Okay. Have you observed any sex or disease stage specific differences in the PERK NRF2 axis modulation or response to the treatment with TUDCA? And as for the timeline used for the mouse model, aren't the 3 and 6 hour MTTP too short for assessing ER stress?
Okay, thank you for your questions. First, unfortunately, no, we only use the male mice, so we don't know if we would, but I imagine that we would have differences between males and females. And I was hearing Alessandra, and it's very nice that more and more people are doing the studies with both male and female mice, but unfortunately we did not use just male mice, so I don't know the response to that. And regarding the timeline, yes, so here when we designed the study, we were addressing oxidative stress. And basically, we had already observed that we had that huge increase of the JNK activation for oxidative stress. And afterwards, we did this, we get interested in this part of the ER stress. We actually had another model with later time points, so 7 days and 52 days, but we didn't assess the ER stress in that model yet. But it would be definitely interesting to know. I think the problem here with the activation of the UPR is the same as Nikki was referring regarding NRF2. And here we have the 3 branches of the UPR that might be activated at the same time or in just slight different time periods, and they might interfere with each other. And also, so it's very difficult in an animal model to know exactly which time point to target. And I'm sure that there will be fluctuations in the different branches of the UPR, and that will influence our responses. So the ideal thing would be to have enough time points to address this. Thank you.
Thank you, Andrea, for your reply. The last but not least question for Fabio Di Domenico. Why did you use 2 inhibitors of the PERK pathway in the treatment with the EPA, and what did you observe in their comparison? And another question is, do you think that the result you obtain in Down syndrome pathology can be extended to other brain diseases?
Thank you for the question. So for the first part, we used 2 different inhibitors. One is the GSK414 that directly acts on PERK, so inhibits the phosphorylation of PERK and its activity. And the second one, that is ISRIB, instead reduce the formation of the phosphorylated AF2 alpha, so downstream of PERK. We used these 2 different inhibitors because one can also reduce the interaction between PERK and NRF2, because as I show you, PERK can directly phosphorylate NRF2 and induce the antioxidant response. The second one instead works downstream of PERK, so it doesn't interact with the NRF2. So we wanted to see if the mechanism and the effect we were showing were also dependent or not on the modulation of the NRF2 signal upon PERK inhibition or not. So far, for the data we have collected so far, we haven't seen much differences, especially in Down syndrome neurons and astrocytes, because they are mostly regulated by the overexpression of BAC1. So the signal is not really working properly at all at this moment. But we are also collecting data on euploid mice, so normal mice, and we are still checking other signal, other mechanism, other genes that are induced by NRF2, but so far we don't have data to see differences between those 2 inhibitors. As far as the second part, I think that it can be extended to other pathologies. I show you that in Alzheimer's disease, we have an overinduction of the PERK pathway. Also, Andrea showed you that in the Parkinson disease, we have an overexpression of the PERK pathway. So probably there is a mechanism that is pushing the ER stress that is overactivating the PERK pathway, and this becomes detrimental because it can activate pro-apoptotic pathways. We believe that metabolic alterations are one of the driving mechanism of this. So this is why we are trying to understand with IPA, and now we are also performing high-fat diet on Down syndrome mice. We believe that metabolic alteration and brain insulin resistance can also be involved in the induction of the air stress. What is, of course, difficult is to understand the window of a treatment, because in Down syndrome, everything occurs very early, because we have an early alteration of those pathways associated with triplication of chromosome 21, while in normal population and then develop age-related neuropathologies, this occurs later, and we need to find when this happens and find the right windows maybe to use inhibitors to try to reduce the activation of those pathways.
Okay, thank you, Fabio. There is other question among the speakers. Ok, quindi thank you so much the speakers for the interesting talks and clear answers. Thank you everyone for participating to this webinar. Thank you Antonio to lead this webinar with me. I hope this webinar has been informative and helpful, and I'm looking forward to keep in touch for next events.
Thank
Good
you.
to see you. Good
Thank
to
you
see
so
you,
much.
Fabio.
Thank you.
Thank
Fabio,
you.
Bye.
good to see you.
Bye-bye.
Bye.
Bye-bye.
Bye, Nikki.
Bye bye.
Bye.
Bye. Grazie.